Welcome to Uncovering Genetic Disease. You're watching Episode 1, Uncovering Fabry Disease. Did you know that there are over 6,000 diseases linked to genetics? Fabry disease is one of these diseases classified as Mendelian, or simple genetic diseases. It's an X-linked lysosomal disorder resulting from the deficiency of a protein called alpha-galactosidase A, which is encoded on a gene called GLA. Symptoms are varied, but can be classified based on the type of Fabry disease that you have. There's type 1, which is called the classic phenotype. This is the more severe case. Some common symptoms include acroparesthesia, which is just a fancy word for a burning sensation in your fingers and toes. There's also angiokeratoma, or red to purple bumps on your skin, usually in the arms, chest, and thighs regions, that are caused from swollen blood vessels. Cardiac symptoms are common, such as left ventricular hypertrophy, or a thickening in the part of your heart called the left ventricle. This thickening makes it harder for your heart to pump blood. Another common cardiac symptom is heart failure, which can be caused from left ventricular hypertrophy, or from another condition called hypertrophic cardiomyopathy, which is a swelling in the heart muscle wall. Finally, kidney failure is another common symptom in type 1 Fabry disease. Now, type 2 has a milder onset. It can be categorized into two main variations. There's the cardiac variant, where patients experience cardiovascular symptoms like arrhythmias, or irregular heartbeats, and the LVH and HCM that we saw in type 1. There's also the renal variant, where patients experience symptoms related to kidney failure. Now, I dropped a lot of terms here, so let's break this down step by step, starting from the beginning. In 1898, two dermatologists named Johannes Fabry from Germany and William Anderson from England, independently but somehow at the same time, described a brand new condition that they decided to call angiokeratoma corporis diffusum. They observed patients with unique angiokeratomas, or those red to purple lesions on the skin that we talked about previously. And fun fact, while the disease is sometimes called Anderson-Fabry disease, we most often just abbreviate it to Fabry disease for unsure reasons. Fast forward to 1947, and researchers discovered that this disease is hereditary, or based on genetic factors. And it wasn't until 1965 that they understood how this disease was inherited, the X chromosome inheritance pattern, on a gene called GLA. But what exactly does X-linked inheritance mean? And why do we even care in the first place how the disease is inherited? Well, understanding the pattern of inheritance actually helps physicians make better and more accurate medical diagnoses. And for Fabry disease, early screening and preventative treatment can be extremely helpful for reducing those severe symptoms like organ failure. So let's dive into the workings of X-linked inheritance. This chart, called a pedigree, maps family history. Squares represent males, while circles are females. The top row are the parents, and the bottom are their children. The important thing to know to understand this chart is that males are XY, while females are XX genotypes. Y chromosomes are only in biological males, so that means that the sons inherit a Y chromosome from their father only, and either one of the two X chromosomes from their mother. Females will inherit the one X chromosome from their father and one of the X chromosomes from their mother. Alright, now that we have the basics down, let's see how this plays out. So let's say that the father has Fabry disease. His son will be unaffected, since the mother's X chromosomes are unaffected. His daughter, though, will have half affected and half unaffected chromosomes. We call that a carrier. But if the mother has Fabry disease on both her X chromosomes, her son will certainly have the disease. Her daughter will have a 50% chance of displaying the disease. Here's a quick recap. And while this seems like a fairly straightforward process for deciding who will have Fabry disease and who won't, things are never that easy. See, there's this phenomenon called epigenetics. It's a process that causes changes to gene expression without ever changing the DNA coding for those genes. There are two main types that throw a wrench in how Fabry disease progresses. The first is called methylation. These are small tags to DNA that make the sequence harder to make into a protein. The second is called X chromosome inactivation, or XCI. Remember how I said females have two X chromosomes while males only have one? Well, having two X's is too much. So in every female cell, one X chromosome will be randomly silenced to keep a balance. And while these inactivation patterns are random, they can have a huge impact. They're important for regulating Fabry disease, is up for debate though, so more research is needed. Alright, let's check in on our progress in understanding Fabry disease. We know it's a Mendelian genetic disease that follows an X-linked inheritance, but we're still missing some key facts. So, it's time to address the protein in the room. Meet alpha-galactosidase A, better known as AGAL. Fabry disease is caused by the inactivation or deficiency of this enzyme. While its name might make you think it's part of an intergalactic spaceship, AGAL is a protein found inside the lysosomes of your cells. It's here where AGAL has many roles, including facilitating proteins to bind, promoting reactions or catalytic activity. It has hydrolase activity, which is cutting smaller molecules into even smaller ones, and then it can also signal receptors. And while all these functions are important and can help explain why Fabry disease is one that causes such widespread complications in our bodies, our main focus today is on AGAL's hydrolase and catalytic capabilities. AGAL's job is to catalyze or push a reaction called sphingolipid metabolism. Most importantly, AGAL takes a sphingolipid called global triosylceramide 3, or GB3, and cuts it into two distinct compounds. In Fabry disease, AGAL is non-functional or underexpressed, so GB3 piles up in the cells, and GB3 is our disease-causing culprit. But why does AGAL suddenly stop working? To put it simply, it's because of mutations. More specifically, the issue is due to mutations on a gene called GLA. GLA is a gene on your X chromosome, and it's the home of the code that tells your cells when, where, and how much to make AGAL. There are hundreds of mutations that can change this code, causing mistakes when making AGAL. These mutations are often private or unique to each person. They can occur in one of the 7 segments of protein coding regions, or exons, and some of these exons have higher rates of mutations, like exon number 2, while other areas have observed none, like exon 4. But what exactly do scientists mean by mutation? And how does it change the protein when it happens on a code that's far away from it? Well, a mutation is a disruption in the protein code, or in the code that monitors that code. In our Fabry disease case, mutations on GLA usually happen in one of 3 major forms: missense, nonsense, and splice variants. DNA is eventually made into a protein by a series of steps that won't be covered in this video. Just know that changes to the DNA lead to changes in the protein through these steps. In our first mutation type, called a missense mutation, a change to a single unit or nucleotide of the DNA causes a change in a protein unit or amino acid. The AGAL protein is no longer the right AGAL protein. The second mutation type is called a nonsense mutation. Here, a single nucleotide change leads to a shortened version of the protein. Finally, a splicing error happens in the step between making DNA into protein. Before the official protein sequence can be made, the DNA is made into a draft called mRNA. Here it must be edited before it can be made into protein, and sometimes the editor makes mistakes when revising, so you end up with a protein that just isn't quite right. Now, each of these mutation types can cause a different consequence for AGAL that prevents it from doing its job, and it's actually better to talk about them in terms of how they affect the protein's function. Prominent Fabry disease researchers Garman and Garboxy classified them into 3 main disruptions to help explain this. See, AGAL has 2 segments, or domains, that are responsible for its activity. Where a mutation happens in the protein can affect how severe the Fabry disease may progress. In the first category of mutation, the active site is disrupted. Typically, the active site is the place where our disease-causing molecule GB3 is cleaved. However, when a mutation is present, something prevents the active site from working properly, and GB3 can't be cleaved. In the second category of mutation, buried residues, or sections hidden deep in the protein that help maintain its structure and function, are disrupted. And finally, Garman and Garboxy called their third category disruptions in interactions. Either the domains or the protein units are unable to interact with each other. And depending on where the mutation affects AGAL, it can have a huge effect on how severe Fabry disease will present in a patient. Now, with all these incredible and intricate mechanisms to the science, you may be wondering how in the world scientists even figured all this out. And that's through a scientific discipline called genomics. Genomics is the study of all genetic material in an organism, including the structure, expression, interactions, comparisons, and functions of genes. It's often coupled with studies in transcripts, or transcriptomics, and proteins, or proteomics, and epigenetics, which is something we covered earlier. Through genomics, scientists can classify other organisms that share similar genes and proteins with humans. For example, the GLA gene that's mutated in Fabry disease is highly related to, or homologous with, GLA genes in rats, dolphins, and plants, to name a few. Unfortunately, or maybe fortunately for these organisms, they don't show any development of Fabry disease even though they have the GLA gene. This leaves scientists to take measures into their own hands. Scientists will alter the DNA of model organisms like rats or mice to induce Fabry disease. They can knock out the GLA gene, but even then, mice will not show any symptoms because there's no buildup of GB3. In order to make a mouse that shows symptoms, researchers decided to cross a knockout mouse with one that had been inserted with a human gene to produce GB3. These transgenic mice showed signs of kidney failure after only 3 weeks. But when they're given a human AGAL enzyme, the symptoms were improved. This breakthrough discovery led scientists to develop an idea for administering treatment in human patients. And speaking of, let's talk about some of these current methods for treating Fabry disease. The disease has no known cure, so delaying the onset of severe symptoms like organ failure is usually the best method of action. Fabry disease progresses with age, so early screening and prevention are important parts of preserving quality of life. The most common preventative treatment is called enzyme replacement therapy, or ERT. This is where an AGAL enzyme is inserted into a patient to make up for their insufficient AGAL activity. This way, GB3 can be cleared, and the symptoms can be reduced. The FDA has approved one called agalsidase beta, and it's been shown to help improve renal and cardiac function and improve quality of life. Pharmacologic chaperone therapy is another commonly used treatment. Chaperones are molecules that help stabilize proteins that have disrupted interactions, just like what we saw with AGAL. There's a drug called migalastat that's been shown to help patients that have the missense mutation on their GLA gene, but these results can be variable. Finally, there are new emerging therapies that target the enzyme that makes GB3 in our bodies. While the other treatments are aimed at promoting the breakdown of GB3, these therapies try to stop GB3 from ever being produced by silencing an enzyme in our bodies called GB3 synthase. Replacing someone's mutated GLA gene through the use of a viral vector is another currently undergoing clinical trial treatment. Remember to always ask your physician for guidance before exploring any treatment option. We've done it! We've uncovered Fabry disease. Let's review what we've discovered. Fabry disease is on the X chromosome and caused by mutations to a gene called GLA. The protein on this gene, called AGAL, becomes deficient and leads to a buildup of toxic GB3. While symptoms are varied, there are a few that cause great concern, like heart and kidney failure. Fortunately, some treatments can help delay the onset of these symptoms, like enzyme replacement therapy and other emergent therapies. Finally, the only reason we understand so much about this disease is through the study of genomics and genomic research. Next time you hear the word gene or genomics, remember how powerful this science can be. I want to give a special shout out to IB 533, the course in Human Genomics and Bioinformatics, offered through the University of Illinois, and to the course instructor Joanne Manister for inspiring this project and teaching me how to navigate genomic data. Thanks so much for listening. If you enjoyed this video and would like to see future content like this, drop a like and subscribe, and leave me a comment on what genetic disease you would like covered next.